Well, it's brilliant to have this opportunity to share with everyone some of our work, which all began with this amazing lady, Rushna Baichi, more than 4 decades ago. If you're interested in one of the most absolutely remarkable stories of resilience in science, please look up her name. You'll discover that among her many achievements, Professor Baichi was elected to the National Academy of Medicine in 1995 for the quote, development of the first digitized anatomy atlas of the human brain and matching algorithms for CT, MRI, and PET scans. Now, these developments cited by the National Academy of Medicine were preceded by another set of events at Penn that would revolutionize the imaging of human brain function. It was at the Cerebrovascular Research Center at Penn that positron emission tomography, or PET's potential for quantifying regional cerebral functions such as blood flow and volume was first established. The radiotracer labeled deoxyglucose, or FDG, developed by Martin Rivich and Abbas Alavi and co-workers, has been hailed as the quote molecule of the 20th century for its unparalleled and unique impact on the evolution of the field of nuclear medicine. The emergence of FDG PET prompted the need for new analysis methods, and this is what resulted in a collaboration with Rushna that gave rise to the work on elastic matching and atlases. In particular, a method was needed to anatomically localize the activation image in a PET scan in order to identify the cortical networks observing the cognitive processes and brain functions under examination. In other words, they had these incredible pictures, as you see here, for the first time of brain metabolic activity as it corresponds to regional glucose uptake, but they needed to know where exactly in the brain this activity was taking place in order to fully make sense of what they were seeing and measuring. It's no different than, say, having a heat map of new disease cases in the recent past. The image or data becomes much more useful when we have more precise information about where exactly the hotspots are located. Here, for example, which state, which city, or even county. And to address this problem, Rushna worked with his student Chaim Breuth, leveraging the fact that to a certain level of detail, the anatomy of normal individuals can be thought of as quantitative variations on a common underlying qualitative plan. In other words, we all effectively share a common anatomical or bodily blueprint. And now this being a virtual seminar, I can't verify by just looking around that all of you who are attending this talk online indeed have 2 eyes, a nose, a rough symmetry midplane, and so on. But I'm going to assume there's a very high likelihood that that's actually the case. And so at the same time, there are obviously differences among us, but we can think of these differences as representing variations on this shared blueprint or template. So my nose may be a little smaller or a lot bigger than yours. My face may be rounder, and so on. Rushna proposed that a natural way of using this knowledge about human anatomy, the face in this particular example here, is to imagine that a given individual's anatomy or face is a warped or spatially deformed version of some template or reference anatomy or face in this case. If this warping function could be calculated, anatomical features or structures can be identified just once in the reference anatomy, and then automatically identified in an individual through the warping function. And things can get even more interesting because we can annotate our reference anatomies with additional kinds of information as shown here. In other words, what Rushna and Chaim had conceived was the first ever digitized atlas of anatomy. The fundamental problem then in this approach to localizing anatomy is to first define the class of admissible warping functions or transformations, which has to be broad enough to enable the reference anatomy to fit all subject anatomies. And second, to develop efficient automated algorithms for the calculation of the appropriate transformation. Now, in developing what became to be known as elastic matching, Rushna and Chaim sought out a method that would optimally balance the similarity induced between the registered image pair with the amount of deformation to the transform image required to achieve the registration result. This reference to the kinematics of the solution was made explicit in their formulation of the problem of matching two images as one akin to the physical process of manipulating and applying forces to an elastic version of the object depicted in one of the images so that its deformed configuration resembles the target object in the second image. The appropriate forces or loads are derived from a potential function, the similarity term in image registration parlance, that provides at each point of the object under deformation a measure of the quote similarity or correspondence between that point and any of the points in the target object. The first object is deformed in this way until an equilibrium configuration is reached, or equivalently until the total potential energy of the system is at a local minimum. By choosing the elastic strain energy as the measure of deformation, the overall cost or objective function for the image registration problem becomes the total potential energy, and the elastostatic configurations, which locally minimize this cost, represent solutions to the corresponding elastic matching problem. And these groundbreaking ideas developed more than 40 years ago brought about a paradigm shift in medical image analysis from slice-by-slice 2D processing to fully three-dimensional analysis. A paradigm shift that was a precursor to active contour and physics-based deformable models that later became pervasive in the field. And here are those ideas implemented for the first time in code and applied to very early CT or X-ray, a computer tomography study of the brain. Why CT? Because MRIs weren't yet available routinely at that time. And a more contemporary version of the same application that I was fortunate to introduce as a PhD student of Ruchna's, this time using MRI, which has become the preeminent modality for in vivo imaging of soft tissue. Results shown here from our group represent the efforts of a good number of people, in particular my former trainees pictured here, Brian Vance, Gary Zhang, Phil Cook, Jeff Duda, and Paul Yushkovich. These developments in atlas-based segmentation or parcellation of images into anatomic regions of interest allows for an automated approach to the regional quantitation of image signal values, as well as morphology, that remains perhaps the most popular method for hypothesis-driven analysis of medical imaging data. And shown here is a typical example of this sort of region of interest-based study, where in this instance, differences in brain region size were not only contrasted between patients with schizophrenia and healthy individuals, but further correlated with age, which allowed us to test a theory that's been proposed of disordered brain maturation in this disease, that if true would provide new opportunities for treatment approaches and strategies. This work, among others, was led by Abraham Dubb, who was one of our earliest clinical fellows at the lab and has long since returned to the clinic and to taking care of patients. Now, once this capability of taking two or more images and registering their fine details was established, a really powerful tool was unleashed. It provided, for example, a way for changes or differences in morphology to be detected and, more importantly, quantified. This is because the registration or correspondence transformation that connects the images directly tells us in quantitative form how the object structure changes or is different between its representation in the different images. And you can see this in the paths that points within the circles shown here travel along as they are transformed into their corresponding positions in the square. Note how these paths or vectors point outwards, indicating that the smaller circle must expand in order to fill out into the larger size of the square shape. You can see as well the refinements of these paths along the boundaries, which quantitatively inform about the differences in the shape between the circle and the square. Now, what's shown on the animation on the right-hand side is the analogous transformation between a chimp and human brain. However, if I were to draw out the paths that points take for this transformation, however, they would be curved instead of following a straight line. They're actually flows. And if you were to squint your eyes and look carefully enough, you might be able to see this effect within the frontal cortex as it's transformed from its chimp appearance into its human configuration. Now, this curvedness is a feature of the diffeomorphic property of the transformation solutions or flows, if you will. We now compute a property that derives from exactly implementing Rugina and Heim-Broit's original continuum assumption. And what's exciting about this development is that, among other things, it's led to a rigorous formulation of a distance or length metric defined over the transformations of diffeomorphisms. And what this translates to in practice is the availability of a quote unquote yardstick now for anatomy that allows one to properly measure the distance between two instances of the same anatomy. And you can imagine this is hugely important because the existence of this proper metric allows us now to compute statistics on organ anatomy and perform morphometry. For instance, to calculate the average brain configuration of a group of individuals, as shown here in work that was carried out with a former mentee, Yanel Lebrucker. We're able to compute principal components, other statistical measures, and so on. This is really at the cutting edge of computational anatomy and is a really very active area of research as well as application development. For example, the chimp-human brain transformation I showed in an earlier slide was part of a collaboration with Tom Schoenemann, a physical anthropologist formerly at Penn and now at Indiana, who remains interested in questions surrounding human brain evolution that can potentially be informed by knowledge of exactly how human brains differ from those of our closest ancestors. So to compare across species, the average brain configurations are calculated and used to compare shape distributions between the species. In particular, brain region of special interest is the prefrontal cortex involved in planning complex behaviors. Their analysis confirmed that this region occupies a proportionately larger part of the brain of Homo sapiens than it does in pan troglodytes or chimps. The identical methodology can be used to parameterize morphological variations across human health and disease, as for example, in the study here of frontal temporal lobar degeneration and Alzheimer's disease, where because these dementias can be confused clinically, there's a good deal of interest in establishing new neuroimaging biomarkers that can help in distinguishing these two disorders. And another way to support disease differentiation is to examine the time course of disease progression. And here again, we highlight the critical role that image registration plays in medical image analysis, namely its use in registering a sequence of longitudinally acquired images to capture an estimate of the annual atrophy, in this instance in a group of frontal temporal dementia patients, where loss of gray matter is evident in the frontal and temporal lobes, as shown in the yellow red scale here, with relative sparing of the occipital lobes as a hallmark of this dementia. The same annualized atrophy result is shown animated here on the right-hand side with respect to the average brain configuration derived from the patients. And what's visualized is the average amount of gray matter lost in just over a year in our group of patients. I hope it isn't difficult to see how these kinds of longitudinal studies can have an important role in not only improving our understanding and diagnosis of disease, but also in the treatment in terms of monitoring and determining therapeutic efficacy. This work is just a small part of a decades-long partnership that our lab has had with Murray Grossman, who directs the Penn Frontal Temporal Degeneration Center at the medical school. Now, I mentioned earlier that our correspondence mappings are now optimized over the diffeomorphisms. These are just a set of differentiable maps with differentiable inverse. In other words, the topology of the domain, in this case, the image that's being transformed or normalized, is maintained or preserved by the transformation mapping. This is important because our labels then are preserved when they're mapped from, say, an atlas to a subject instance. These diffeomorphic maps are generated by integrating a time-dependent vector field. This is a velocity field effectively over time, starting from X, the identity configuration. And the idea here is that by ensuring the velocity field is smooth, the displacement mapping we get by integrating the velocity field will themselves be differentiably smooth. In other words, diffeomorphisms. There are standard constraints on the optimization problem. The first is that at time 1, the diffeomorphism should map image I, shown on the left here, into the space of image J on the far right. And the second constraint is that the length of feed of mapping should be as small as possible, and this length is described by the integral over time of the velocity field size. So by solving this problem with these constraints, in other words, the registration problem, we can smoothly map image I into the space of J, as shown here. And here we see an animation of a diffeomorphic map in this example between a chimp and human brain that we showed earlier. Each frame in a time lapse of the movie corresponds to a point along the diffeomorphic map. We can get some additional insight into the algorithm by taking a look at how we discretize the problem. As we saw in the preceding slide, the dense in space and time velocity is sampled at a finite set of time points. And looking at the integral, we can see that the initial value of the diffeomorphism is the identity phi x at time 0 is equal to the identity, it's equal to the initial configuration itself. To update the value over time, we simply traverse the velocity field, accumulating the deformation in small time steps until time 1 is reached. Now, as one might imagine, this looks locally at a voxel like a curve integral, and indeed this is exactly what it is, except that it's spatially dense in a continuum instead of at a single point. So for that reason, a diffeomorphism locally over time traces out a curve, which is defined uniquely by its initial value, the identity, together with the tangents of the curve described by the velocity field. So to optimize the diffeomorphism, we basically manipulate the values of the velocity field. It's important to note that this is a very distinct concept from just simply applying a series of incremental deformations to an image, something that's conventionally done to mimic large deformation transformations. But the trouble with this approximation is that it doesn't guarantee that domain topology of the warped or transform image is preserved. It also doesn't assure that a proper inverse mapping will be available. And I'm going to see very shortly why this is really important. Now, there is, however, a quite serious shortcoming of the approach that I've just presented. And in fact, this shortcoming afflicts all conventional registration methods in general. And that's the fact that the formulation and the viewpoint it takes are fundamentally asymmetric. The path from image I to J and the opposite or inverse path from image J to I are actually not the same, meaning that the metric distance between our endpoints, in other words, the total deformation summed in time, actually depends on which image we choose at the reference point. In other words, the direction in which the registration is performed matters or makes a difference. This bias is clearly a problem for the kinds of quantitative applications we're interested in for any of our research or clinical imaging studies. Unfortunately, there is a way to fix the problem and to see this. The idea that was developed as part of the thesis work of my former student, Brian Avance, whose picture is shown here, along with Charlie Epstein, who was at the time in the math department, also contributed to this work. The idea is, again, to generate the mapping phi in the same way as before as the integration of velocity field, except what we do now is we move the endpoint condition to the middle of the path by explicitly representing the component of the diffeomorphic map that operates on image J. And what this means is that the geodesics, or the shortest diffeomorphisms connecting the two images, are now required to be the same. This now gives us a set of symmetric Euler-Lagrange equations that depends on gradients of both images I and J, as opposed to just image I in the original formulation, which is the asymmetry that causes the bias in standard registration. The second important numerical difference is that the optimization can also be carried out symmetrically now. So we not only have a symmetric formulation, but the optimization can also take advantage of this. And the way we do this is we discretize in time the velocity field as we did before, except we then update the velocity field symmetrically from both endpoints with initial phi values, and then generate new phi values by integrating as before the velocity. And this process is repeated until convergence. Now, I've been told sometimes that the method as I've just presented it can be a little bit opaque to audience members. So please don't be too concerned if this is indeed the case for any of you out there. The basic algorithm is actually really quite easy to appreciate when we take a look at the symmetric normalization in action. So in particular, let's consider, as shown here, the sad face on the far left as image I, and the happy one in the middle as image J. I'm using smiley faces here only because it's a lot easier to show the effect of the algorithm with the faces than, say, with brain images. So note where conventionally one image is typically deformed into the other, the sad into the happy, or the happy into the sad. Both images now deform into the mean configuration at time equal to 1/2. And the total symmetric normalization, or the total path between the two images, is obtained by composing the two diffeomorphisms shown here. So rather than going from sad to happy, I to J, or happy to sad, J to I, both cartoons first become sappy. I'm supposed to hear you kind of laugh. And anyway, so to go from sad to happy, for example, you start with the sad, depressed state, and you flow into the sappy state, into sappiness. And from there, you take the inverse mapping from sappy to happy. And the path from I to J now is identical to the path from J to I. And what's even better is that this symmetric registration not only gives us unbiased results critical to quantitative studies, but also results in significantly improved performance, because all of the images now contribute to the calculation, as we saw earlier when the gradients were shown to be taken with respect to both images in the pairwise registration problem, as opposed to with respect to just a reference image in the original biased formulation. This performance gain has been essential in enabling ever more precise and accurate results, currently top ranked in the field, but it's also made possible wholly new applications as well. And one important application that I'd like to highlight here is how the symmetric normalization approach naturally generalizes from pairs of images to define a symmetric population study over arbitrary numbers of images. And an easy way to see this is to note that the pairwise symmetric normalization algorithm that I just described effectively solves a two-mean problem. We saw how we actually solve for the mean configuration, the average configuration when going from I to J and J to I, and then transition with the mean configuration to get from I to J or J to I. Well, a population study simply corresponds to solution of an arbitrary n-mean problem instead of the two-mean problem, where n being the number of subjects in the group analysis. And this is exactly what the generalization provides. It finds an optimal reference coordinate system or template I bar, or atlas, if you will, atlas template given directly by the n-mean configuration, which by definition minimizes the total length of the set of diffeomorphisms VI operating on our subject images JI. And these diffeomorphisms are exactly the population normalizing transformations of interest to us in a population study. Well, I did want to make sure that we had a bit of time for questions at the end here. So let me wrap up by inviting those of you who may have an interest to take a look at our lab's freely available software resources and others to which we contribute that implement everything that I've presented today. Thanks very much.
So yes, I'm Jason Marsh. I founded Flow Immersive 8 years ago. I started my high-tech career at Apple Computer back in 1991, working on speech recognition. So been around a little while at the intersection of humans and computers and alternative ways of interacting with digital information. And so as much as I like to talk, I'm actually going to share screen and talk over visuals, because it's much better to see than just hear me describe things. So what Flow aspires to do is to help people have the best data conversation they've ever had and feel like they're making the best data-driven decisions that they've ever made. So the video is just a set of different kinds of use cases. So you know, mostly from general enterprise work. And what are some of the key principles upon which we've built the system? First of all, it is augmented reality first, but it works very well on flat screens as well. And I'll be doing my live demos on flat screen today to show you that. It is collaborative. So having multiple people in the same space at the same time, where you can see their avatars if they are remote, and you can see their laser pointers if they are in person, so that everyone is looking at the same augmented reality data at the same time, and anyone can select, anyone can point at a dot and say, tell me about this. Anyone and everyone in the meeting can see what you are all engaged with. And the point really is to take us to the next level of how we have, you know, really good data-driven decisions. And I kind of feel that humanity has a few problems that need some solving, and being able to use data more effectively in those meetings where those problems can get solved would be very helpful. For a lot of our use cases, it's in things like consultancies, where The consultants are providing a end report to customers. In financial services, like this one, we're looking at a stock portfolio, like a wealth management situation where you want someone to be able to understand what's going on with their portfolio and the markets without showing them an 80-page slide deck. These decks really negatively impact our ability to absorb information. And that's a key problem. As part of that, the whole system needs to work in an enterprise workflow so that there is a flow editor, which is some of what we're kind of showing here, which is a flat screen environment, browser-based editor that enables you to very quickly wire up your data, see it in 3 dimensions, and then share it widely as you would like based on your security requirements. There's lots of different visualization types. You can kind of get a sense of it. You're just looking at a force-directed graph right at the moment. But between force-directed graphs, maps, and a variety of things that I refer to as alternative layouts, you can actually have incredible number of ways of defining how dots appear in 3D space. And that's, so in some ways, that's what we're doing. I'd like to switch to show some of the, show a quick example of a project that first on video here, and then I'll do a live demo of a project that we're working on for the National Cancer Institute. Let me be more precise. We're working with an organization called Qualitative Imaging, and they do, well, you guys, Andreas probably knows more precisely their products than I do, but the big picture is there are medical experts and imaging expertise, and we're the VR or augmented reality part of the equation. And so if my knowledge of the actual content seems a little bit thin, it is because it is. But with them, we did an SBIR grant for the National Cancer Institute to look at bringing microscopy data into virtual reality and seeing what effects that could have on teams to understand more detailed information. And we got exemplary marks across the board on that. And we have applied for our phase 2 of that grant, and we hope that will be approved in July, and we can dig into it. So this gives you a sense of what the data looks like in augmented reality. I'm going to switch to the live demo to show you what it looks like on a flat screen and talk through some of the information around that. But before I do that, let me just make sure that you can see this, that the sharing is working reasonably well. And Andreas, I'm going to ask you if I'm on the right track in the presentation so far, and there's something you would like me to emphasize or questions you might have.
I think just keep going as you do. I'm writing up questions here as you go. So if you could leave, 5 minutes at the end for questions, that would be great.
Super. I'll give you more than that. I try to be concise in describing what we do, even though I call it an infinite problem set. There's an infinite number of ways to visualize data, especially in 3D, and then how to collaborate on it. And then once we get to the AI piece, it's kind of, we could go on forever and ever. Let me describe this project just a little bit more. So we are in a browser, and it's a 3D environment. There's a pretty different feeling in VR, AR, where you see this tissue sample spread out in front of you. Maybe it's 2 meters big, maybe it's 20 meters big, and you're walking around it, either flying virtually or not. This is a kind of old prototype, and you're seeing some of the resolution. It's not full resolution in this particular demo right here, but this is a tissue sample of I believe it's a liver cancer. And in this first view, we're just looking at biomarker layers. You can pick between, in this particular data set, there's some 48 different biomarkers. You can pick between them and apply a certain color to each of those on this view. So it's a layered view, as well as, basically turn them off with black, but you can see the different aspects here. But in addition to just colorizing biomarkers, which is something that, you're probably very well familiar with, you can also show the amount of the expression of those biomarkers based on the height of a dot off of the cell. So in this case, where each cell has its own biomarker indication, and we can, get a sense of the cell size, but also the expression. Right now, I'm showing CK19. We can swap to, a completely different biomarker, and here you can see particular spikes around CK7. I will defer to you guys to know exactly what CK7 means. In addition to the biomarker layered view, there's also a cell classification view. And in this case, what QI imaging has done is they've gone through and used techniques to identify, what are tumor cells, immune cells, and be able to highlight and turn on and off each cell type and see how things are interacting together. Again, we can hide the overview and just see the cell classification directly, or we can pick between the different biomarkers. And then finally, in addition to that, there is a 3D scatter plot view where you can take any of the biomarkers or the center points, in this case, nuc X, nuc Y, nucleus center point on the sample, and pick between, pick which correlations you'd like to do. This is not incredibly sophisticated in this implementation. It's not, we haven't done a bunch of like machine learning to do advanced classifications or to see groupings and correlations that would be interesting. But for a first pass part of our SBIR phase 1, this was pretty interesting to put a bunch of researchers in the same view and have them be able to walk around, and every click they make, every other researcher sees. And the live demo we did, I think we had people from all over the country looking at it simultaneously. I would like to talk about the AI piece of this because I think it's really huge. And to do that, I'm going to do a demo on climate data. I'm kind of a fan and something I know a lot more about than microscopy, for example. And what we're doing, I would consider our AI implementation to be in progress, ongoing, and beta at this point. But I still think it's pretty interesting. So first of all, we're just looking at global sea surface temperatures over the last 40 years. You can see it's getting warmer. Maybe we'd like to figure out a way to manipulate this visualization in headset without having a big complicated interface. Filter to show me 2016 to 2024. So what we've done is we've wired up first the Whisper API from OpenAI in order to recognize my voice. It looks like it didn't recognize my voice exactly right, but our software decided to do the right thing anyway with the filtration and filter out the data. Why is this important? I think that putting an AI voice interface on top of a headset in a headset environment is really vital to give the end users a sense of mastery and control over their data and information. I think that doing complex AI interfaces, complex UX, I should say, in AR is a challenge. I think it's not comfortable. It's not the way we're used to seeing the world. Sometimes we don't have as much resolution as we would like, or as wide a field of view as we would like in augmented reality. And so being able to use just a voice interface is a very powerful and maybe absolutely necessary mechanism. I'm going to show a different aspect of our AI implementation, but before I do, I'm just going to say that we're looking at land temperature data. This is anomaly data going back to 1850. Each dot is a month, and we can filter on any particular month. In this example, you can see that September of last year was hot. It was freaking hot. This delta between, it was so much warmer than any other September, about 0.44 degrees Celsius warmer than any other September we've ever seen. So interesting to see this kind of data and have, really control over being able to visualize it. But let's have a little bit of fun with this. Put this on a 3D radar chart. So we've trained our AI to understand a variety of different templates in our system. And it will, based on what I ask, in this case, I asked for a specific template, but you could just say, visualize this. It takes the data and figures out which attributes to apply to, which parameters of the visualization. Now time is going, deep into the view, and the distance from the center point is the amount of the anomaly. This is a pretty wild view, just seeing how much warmer last year was than any prior year. This gap here on each and every month, yeah, that's not good. But it's pretty fun to just watch it spin the data into a new view. Okay, I've got one more thing I'd like to show, and then I'm going to break for questions and discussion. This view is, we're looking at the temperatures of the South Pacific. It's called the Oceanic Nino Index. And we can see that our last very strong El Nino was 2016, which until this last year was the warmest year on record. But here's some nice, interesting ways to think about AI. Tell me how this affected Mongolia. So what I'm doing is I'm using the context of the dot that I have selected as the input. So it knows what I mean when I say this. It knows that I'm talking about 2015 and El Nino, and particularly even what period within 2015. And then it's using its general corpus of knowledge to answer the question. In some ways, taking any data set and providing a whole nother level of context. But there's, probably even more important from a researcher standpoint, is not just using that general corpus of knowledge, but also being able to ask directly out of a PDF document. What's the prognosis for El Nino temperatures moving forward this spring? So I have uploaded a document from the Climate Prediction Center, and we have set the creativity down to 0 so that it will only and always answer directly from that document, and it will respond, I don't know anything about your question, if that information is not present in that document. So working with private data sets, of course, which is what all our customers do, and private analysis of those data sets, I think is an important piece of the overall AI puzzle. Moving forward, we are doing a lot of work with the AI, actually writing the Python code to process the data and revisualize it for us, as well as being able to ask questions like, what insights should I be noticing in this data set? And have it provide some valuable information to the user. All right, so that was a whirlwind of information, and I'm totally happy to discuss or take any questions.